This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 269, recorded on July 7th, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Is it really hot there, Michael?
We have swamp weather. This morning I walked out and I was drinking air. It's so full of humidity. I thought I was in St. Louis for a while.
What's your temperature?
It's 91 degrees,
wow.
which I think is, 32 degrees centigrade?
Could be. It's 27 here. It's not so bad. Also joining us from St. Louis, Petra Levin.
Hello. Right now
back.
in St. Louis, it's cloudy, and I would guess it's in the 80s. It was supposed to be almost 100 today, but we lucked out with some cloud cover. But the humidity is high.
Gets hot there and humid, right, in the summer?
Yes, it usually gets very hot and humid. More like we'll have spells where it's extremely hot. It's always humid. But we'll have a week or two every summer that it's up near 100.
Well, we are back in studio. I guess the last episode was was in Washington, right, Michael?
I believe so.
Am I right about that?
No, the last one was in studio.
No, we were here. We were here 2 weeks ago.
Yeah,
that's right.
I mean, but
I lost track of time.
You've been doing too many TWiVs.
Well, I cut back to 2 a week now. We're not doing 3 any longer, so that's good. Yesterday, I finished closing my lab at Columbia, so I'm here most of the time now at the studio. Do you know, Petra, I call our recording studio, which is where I am, we call it the incubator.
Yes, I remember you calling it that.
Isn't that cool? Don't you love that? It just reminds me of
that.
Those, my first incubators were for plates of bacteria and just those metal things with a thermometer sticking out the top.
I think it's better than calling it the warm room.
Yes, you don't. No warm rooms.
We had a warm room at Columbia. We have it on my floor. Yeah, we have a warm room.
Yeah, I'm very lucky. We have a warm room, so we have a shaker, platform shakers in a warm room. I feel very, very lucky to have that.
You are indeed.
We do. We have a platform shaker. I left everything behind. I have the big floor shaker that you open up the top. I have one of those. I left it behind. It's all for whoever wants it.
I hope somebody takes it. That's good stuff.
It's good stuff, and it's expensive to replace today.
Yeah, I know. I left a lot of stuff in the lab that I couldn't move, and I hope someone takes it. But there is someone else moving into part of the lab, so good riddance. I will do the rest of my years doing teaching of this sort and teaching students and teaching via podcast, which is very good with me. I love it. And today we're going to continue our teaching. We're going to start with a snippet from Petra on a paper that we discussed on episode 261 with Mark Martin, but that was a preprint, and now the actual paper has come out in Science, and there's a commentary by Petra. So we thought it would be a nice addition to that episode to have Petra sort of summarize her thoughts. So take it away.
What I really want to know before you get started is who did the graphics for your...
Oh, she is wonderful. I actually emailed back with her back and forth, and we had a very interesting discussion about, not long discussion, but a little bit about how you get into this. And she said she'd be happy. My daughter was very interested in the image and loves art, but this idea of, so the picture that goes with my commentary is phenomenal. I think it totally makes the commentary. Actually, it's a cross section of this giant bacterium. And the artist is Kelly Holoski, and she has a website and has fabulous, we could add it to maybe the show notes, really beautiful pictures on the website.
Tell me your name again, Kelly.
Kelly, Kellie, Holoski, Holoski. And
Okay.
if it's not easy to find, I can send you the link. And she has really beautiful images. But I think this is, when
There
I
it is.
first started graduate school, every department, biggish department, would have their own artist. And we had an artist. But then with PowerPoint and online graphic, with doing our own graphics, we lost that. But I think we lost, I can make a rudimentary picture of pretty much anything, but I think my skills as an artist are very poor. And so to have somebody do a figure like this is really a treat.
Well, that's why I wanted to revisit this paper, because I think the art actually will help the intro micro student really appreciate the significance of what your commentary talks about and then what the primary paper also says.
Oh, 100%. I think this figure, you could just put it up and then put a picture of a eukaryotic cell next to it and really do a comparison of what's allowing this bacteria to be so large. But
Her website has all her previous science illustrations on it. It's quite nice.
Oh, yeah, they're amazing. They're beautiful. They remind me of like these images people would draw, even in the 19th century, these incredibly intricate, beautiful botanical images.
Hallucinations in mice. How about that?
So we'll put that into the show notes.
Yeah, we will. You could go visit Kelly's website. It's very cool.
Yeah. But I guess we should talk about
the data.
So, well, I'm going to talk about, I think since Mark really went through the data on his show, and the preprint isn't really that much different, very different from the actual publication. And I encourage everybody to read this paper. It's called, the title is A Centimeter Long Bacterium with DNA Contained Metabolically Active Membrane-Bound Organelles. And it has a good number of authors, but the first author is Jean-Marie Voland. And the last author is Shailesh Date. And it's a really nice consortium of authors who bring different skills. Actually, one of my colleagues who would be great, I think, to have on TWiM sometime, Esther Ongert, is on here. She's an expert. She works on another extremely large bacterium from a totally different family, Epulopiscium fischelsoni, and she's an expert on how these bacteria reproduce. And she's amazing. But again, this paper talks about the identification in a mangrove swamp in Guadeloupe of a bacterium that is a centimeter long. And actually, I think Carl Zimmer's article in the New York Times indicates like the way to think about this is it's as long as an eyelash. So obviously that's something you can see. So it means, and the title of the commentary I did is, it's a bacterium that is not a microbe. And I've been thinking about just that alone brings up like, are we microbiologists if we study a bacterium that is not a microbe? Maybe it's better if we term ourselves bacteriologists, parasitologists, and virologists as opposed to microbiologists generally. Then we don't have to worry about these. But I think, the history of bacteriology and microbiology is all first seeing things using a microscope that you couldn't see before. So I think that already is a philosophical question.
Let me ask you, I asked this of Elio once. I asked him if a virus was a microbe. He said, sure. What do you think?
I think sure is a very good answer. I mean, For the most part, I don't think there's a single virus, even the giant viruses we can see with our eyes. So they are microscopic, and the definition, which is from, textbook definition of a microbe, is an organism that you need to
beyond the resolution
of the human eye. You need a microscope.
Okay, good. Because I've spent my career in a microbiology department, and now my company is called Microbe TV. So I'm glad it's a microbe.
I mean, I think, there probably are a lot more of these very large ones out there, but I think if you have a normal curve of sizes of things, bacteria, viruses, and parasites, and archaea, and fungi, they're going to fall. Well, obviously fungi aren't so great because many of them are multicellular, but they all would fall into a normal curve. And these would be one end of the normal curve of the single cells that are big enough to see.
Right.
It's fair. Good. It's close enough. So this microbe, just to summarize, is, again, about as long as an eyelash. It's a member of the large sulfur bacteria. And these bacteria many of them are quite large. The previous record holder, Thiomargarita namibiensis, is a round a member of this LSB, large sulfur bacterial cluster. And a sort of general feature of them is they use sulfite as the electron donor and nitrate as the electron acceptor. They haven't shown this in this organism, but it is probably the case based on the genome and the structure. And in this group of bacteria, they tend to have a sulfur inside as a donor in the cytoplasm, these balls of sulfur. And they have a giant vacuole. And this is my favorite part. They're giant vacuole full of nitrate, which is like we use oxygen as our terminal electron acceptor. So I think of it as like a giant scuba tank. Somebody once described it that way to me, and I love that image, is they're carrying around the nitrate. And some of these circulate between the sulfur-rich and nitrate-rich parts of they're usually live in water, aquatic environments, sulfur-rich and nitrate-rich parts of the system. These guys, the Thiomygarita magnifica, is attached to the mangrove so that they don't seem to circulate, but they must have access to both the donor and recipient. And they're probably using fixed carbon. They're not fixing carbon. So they eat carbon containing organic molecules like we do, but they use inorganics to generate proton motive force and make ATP. And that's true of a lot of bacteria. And I think, there's endless variety. And if I get up on a soapbox, I think we should be teaching how proton motive force and how it's generated in things first from a bacterial perspective as opposed from a mitochondrial perspective, because that would make so much more sense. But in any case, okay, off my soapbox. So in this, the paper is beautiful, and they go through a lot of things to show that it is microbial. That is a bacterium, not microbial. It's a bacterium. And this is true of actually previous work on Thiomygarita namibiensis and Epilopisemium fischelsoni, these other giant bacteria. You want to prove that it is bacterial. And that's actually fairly straightforward to do. You can just sequence the ribosomal RNA loci, and you can show it fits into, in this case, the group of the LSBs. And then they try and get an understanding of its metabolism and how it reproduces. And it has a couple really interesting features. It has these, most interesting, I think, is these what they call pepins, which look like they're membrane bound. And it's actually, there's a really nice piece by Christoph on Small Things Considered about whether or not these are actually independent membrane bound vesicles or whether they're actually attached to the inner plasma membrane. And it's actually impossible, I think, to tell from what they have whether that's true or not. But nevertheless, inside each pepin, which is for the most part almost completely, it looks almost independent, is encoded a copy of DNA. And then the DNA is looks to be simultaneously transcribed, so made into RNA and then translated into proteins. And one of the common features of all large bacteria is that they have multiple genome copies, kind of distributed genomes. They're polyploid. Some of them have hundreds of thousands of copies. These guys are more in like the 40 to 60,000 copies per cell. And that allows you to have your DNA kind of distributed around, in this case, the periphery of the cell, and have simultaneous transcription and translation. So basically you can make the RNA and protein you need where you need it. So you don't have to transport it across long distances. But I think that's one of the most kind of amazing things about this guy, is that it seems to have what we would think of as more like a traditional organelle, or another way to think about it is little tiny bacterial cells inside this monster cell.
It's the original just-in-time delivery mechanism.
Exactly. It is the original just-in-time. So I think one of the, things that I want kind of high 30,000-foot view here is, I think this, again, is another example of what we were taught or how we teach about bacteria and prokaryotes generally, is that they do not have membrane-bound organelles, right? The difference between eukaryotes and prokaryotes is always the the prokaryotes, the bacteria and the archaea, don't have membrane-bound organelles. They just sort of have the DNA inside the cytoplasm, and that limits how big they are because they can't transport things. And it also, they're limited because they don't have membrane-bound compartments. Their surface area to volume ratio makes it difficult to get enough nutrients from outside in and to get waste product out. And I think we should know better at this point, but clearly we don't. I think eukaryotes have solved this problem. There is a limit. There are some very large eukaryotic cells. I think, classic one is like an ostrich egg, or there's some very big algal cells. But bacteria have also solved it. They've just solved it differently. And I just want to go through the different adaptations that these guys have that allows them to be large. And again, the adaptations are what appears to be a very invaginated plasma membrane is one of the adaptations. This polyploidy, this many, many copies of the genome and just-in-time delivery with a distributed genome all around the cell. And then a third one is finding a way to, in this case of this cell and the large sulfur bacteria as a group, having this membrane bound or this vacuole inside that essentially presses the cytoplasm up against the surface of the cell. So the cytoplasm volume is actually fairly small and the cytoplasm is fairly thin. And so that allows for exchange of things, nutrients and other small molecules into the cell and waste out of the cell without having to transport it across vast cytoplasmic distances without the kind of transporters that exist in eukaryotic systems. And then having this invaginated membrane also helps increase the surface area to volume ratio. And then the distributed genome in just in time. And again, this is a theme that comes up again and again in these, as we identify more and more of these larger bacteria. To me, it's sort of, when I first saw this, I was surprised to see a bacteria I was amazed to see a bacteria that large. But when I thought about it, I thought I shouldn't be surprised because these adaptations seem to be a way of allowing cells to essentially overcome these physical differences that we were taught are why bacteria are small. But these physical issues actually apply to eukaryotes. They just have come up with, or through evolution, different adaptations that allow them to increase size. So bacteria and eukaryotes have both solved this size problem. They've just solved it differently.
Well, one of the most intriguing pieces that you had in your commentary that I don't remember from the original paper is the fact that the surface of the bacterium has nothing growing on it. And you point out in your commentary that the surface of this bacterium could accommodate up to 625,000 E. coli. And the fact that the surface doesn't have any biofilm attaching to it, if you will, a bacterial parasite on a bacterium, it is really remarkable because the biotechnology applications of the material that this microbe is made out of could actually give us a new medical grade catheter that wouldn't grow staphylococci or streptococci on it.
Right. This is actually remarkable. And that might be another adaptation that allows them to be so big, because obviously if you can have stuff growing all over you, that's going to kind of suffocate.
Slow you down.
So it turns out, though, I think it's, from what I can tell from reading, and I think what the authors say, is that the genome encodes a lot of non-ribosomal biosynthetic operons, basically operons that encode enzymes that can synthesize maybe peptides, or they can synthesize other kind of molecules, ketides and things like this, that actually have, some of them, antimicrobial properties. And in fact, many of the antimicrobials we get actually come from operons similar of biosynthetic operons, similar biosynthetic operons that are in the actinomycetes, specifically in the streptomyces. And these organisms, the streptomyces, which I always like to remind people, give dirt the smell. They make geosmin, which smells like soil. these guys, the streptomyces, so they make streptomycin, and a lot of the sort of precursors for drugs that we use, antibiotics we use, come from these. So this bacteria, Thiomargarita magnifica, which is in a totally different group of organisms, not even the same, seems to also have, possibly by a horizontal transfer, acquired a lot of these biosynthetic operons for making different molecules with antibiotic or antimicrobial properties. And I think the authors would like to say, and I agree with them, that that's probably how it keeps its surface so pristine. Because, yeah, you easily could form a biofilm on this thing. It's huge, and yet nothing forms. So I thought that was really fascinating. But whether that's a necessary adaptation for size or not, it's the first time it's been seen. If we see it a few more times and really when we discover more of these giant organisms, I think it would be another list we can add to the adaptations necessary to be this large size.
Petra, what's the function of having so many genome copies along the length?
Right. So it's like Michael was saying, it's the idea is that if you have a genome if you have one genome in the middle of a cell or two genomes in the middle of a cell, if you're only a few microns long, like E. coli, you don't have to transport things very far because you're only half a micron wide and a few microns long. But it takes hours to transport things by diffusion in such a big organism as this. And so in this case, by having genomes distributed all along the periphery of the cell in these pepins, these membrane vesicles, that allows the cells to make proteins where they need those proteins.
And the other thing that Petra pointed out earlier is it's establishing a proton motive force gradient. And a lot of biosynthesis is done via PMF. And the fact that the PMF is distributed along the entire length of this bacterium enables it to have energy. It's not like they're transporting energy currency in the form of ATP. It's the PMF that is actually responsible for generating the reducing equivalents that can actually make more protein, more cell mass, more everything. And the fact that they're able to, if you will, have your own Bitcoin generator right there on the spot, printing money. And you got the sulfur grain next to the pepin that's making the protein. That's why that drawing is so incredible. And the fact that the vacuole is just full of the electron acceptor that you can literally just dump your trash into this river vacuole called nitrate and it becomes nitrite. I mean, it was just, the drawing is what really got me so stoked about it, because I remember talking about it with Mark and, you know, I wasn't that excited, but when I looked at the picture and really studied it, I'm going, this is wow.
Yeah, it really is amazing. But again, I think the pepins are a new feature, and the ATPase, when they localize the F1FO ATPase, is sort of the thing that's in the membrane that can take advantage of proton motive force to generate ATP for generating energy, for doing everything. Those are also distributed in this membrane around the pepins and also this other invaginated membrane in the cell. And so that even allows them to take advantage of all this PMF they're generating to do that.
So the other advantage of having so many copies of the genome is the daughter cells just bud off the end and they automatically get the genome. You don't have to bring it anywhere.
Exactly. Right? You don't have to really, well, at some point you have to segregate and the pepins have to divide, and they haven't really looked at that, but the daughter cells pinch off one end of these long filaments and they always contain these pepins, which are presumably containing the genome. They seem to contain the DNA in the cell is all in these pepins.
So I wonder what would happen. The mutation rate is low, but because this is DNA, but eventually you're going to have genomes with changes throughout this organism. I wonder how that's dealt with.
Right. That's a really good question. I mean, this is a question even in bacteria like E. coli, where you don't have complete copies, but you'll have, especially in rapidly growing cells, multiple copies near the origin. And people think that's also important for repair purposes, has been one idea. Here, right, you might have one pepin at one end of the cell that's very different from the other end of the cell. And is that important? Or maybe the pepin, if it's a if it's a catastrophic mutation, my guess is that pepin would go away. But if not, you probably have a lot of genetic variability. You'd have to do, I guess not single cell genomics, single pepin sequencing.
But you can at least dissect it because it's as big as a house.
It is, but the pepins are like E. coli size. So
yeah, they're E. coli size. I'll grant
that's kind of challenging.
That would be an interesting project to sort out pepins and then do genomes and see what you have in terms of variation along the length, right?
That's cool. Yeah. I mean, but again, it brings up a question that's even true, I think, in smaller organisms or average size bacteria, which is how does the cell know to divide in a certain place? And are the pepins at one end communicating with the end that's dividing? It looks like there's pepins near where the cell is dividing to produce a daughter cell. Those might be more active. But again, there are just so many great questions, I think, to ask here. Where are the proteins? Definitely has some conservation of the cell division and cell peptidoglycan machinery. Are those made all over? Is it growing at one end? How is this controlled? Is one end talking to the other end? And if so, how? I mean, it's again, the diffusion rate is too slow. So I think it's
one of the authors that one of the papers that you cite in your commentary is from one of the professors who taught me cell division, Arthur Koch.
Oh, yeah.
And Arthur would, all of the questions that Dr. Levin have just phrased, Arthur was phrasing back in the 1980s, torturing us poor graduate students, these questions before the advent of all this wonderful molecular biology that we have. And he was just like 30 years ahead of everyone else. Did you ever meet Arthur?
I never met Arthur, but as a graduate student, when I was interested in division, I wrote to Arthur and he sent me a whole packet of, and I read about, because there's a lot of biophysics about the pressure in which cells divide. And I think on this scale, it's probably a little different, but how do cells divide? And he had he was really wonderful.
He was a great teacher. And I'm going to use one of his examples when we talk about the next paper here in a few minutes. But he was just so clever, and it was so intriguing to just listen to his lectures and how much thought he put into thinking about, simple things like, how does the DNA know where to go?
Yeah. I mean, he was sort of a first principle guy, and most of us aren't trained to do that. We're trained to gather data and then analyze the data. But he was a lot of his work on division, at least, is really like, let's think of the bacterial cell as a bubble and with this kind of pressure, and how does it, which is a great way of thinking about it, because it kind of forces you to put the other things in context, but it's not how we're trained to do it. So he really was a unique individual in that sense too.
Yeah.
So I guess I ended mine, and I think this is true. And I think about this in terms of antibiotic resistant, the sort of, no matter what we throw at bacteria, they always seem to find a way to become resistant to it. I think this is a different side, sort of a much more fun and less threatening side. But I think, we shouldn't ever underestimate them. And I think too often, even the way we teach about the difference between bacteria and eukaryotes is if bacteria are lesser. I think they're just different and they are equally adaptable, and maybe because some of them grow fast at least, or they replicate their DNA quickly, they are able to adapt to whatever kind of we throw at them. And, if we are throwing at them antibiotics, they'll adapt. In this case, this guy is really big. It's not clear why it needs to be large. Maybe it's a predator situation. But in any case, I think it's just another example of we shouldn't underestimate bacteria.
And these can't be grown in culture, right?
Not yet, no.
That would be good to crack, wouldn't it?
Yeah, I think it would be good to crack. I was reading in one of the popular science articles about it that they might be seasonal. They see them, and then sometimes they go to find them and they can't find them, and then they'll go back and they'll find them again. So I think there is some seasonality. I think it would also just be great to maybe have a systematic search for organisms like this. We can't find them using metagenomics, right? If you just sequence a whole bunch of an environmental sample, you won't know how big the thing is. But I think if we, tried to be a little more careful about testing things we see, in this case, kind of strings hanging from mangroves or whatever, I think we could find there are likely more of these large organisms out there. Maybe one would be easier to grow in culture. So far, I don't think the three, that this one, Nimibiensis, and then Epulopiscium, which is actually a firmicute, totally member of like the same group of bacteria as Bacillus subtilis or Staphylococcus. I don't think any of them have been grown, at least long-term, in culture.
Yeah, it's a very nice story. People are fascinated by big things, big viruses and big bacteria, right?
Yes, I actually learned from talking to, I think it was Carl Zimmer was telling me that the large viruses were originally thought to be bacteria.
That's right.
And then people went back and said, wait a minute, they don't have the features that you would expect.
Mm-hmm.
So.
That's why they called it MIMI. MIMI stood for microbe mimic.
The
MIMI
MIMI virus. How big is the MIMI virus?
The particle is 750 microns in diameter.
That's staphylococcal size.
Is it?
Oh, 750 microns?
No, 750
nanometers.
Nanometers.
Sorry, nanometers.
750 microns would be Epulopiscium size.
750 nanometers and
micron. A nanometer, sorry.
Nanometers. Yeah, nanometers is correct because polio virus is 30 nanometers, right? The actual capsid shell is only 450 nanometers, and the rest are these fiber little would look like hairs basically coming out of the capsid, and they give it the additional dimension. Yeah,
but 500 nanometers is E. coli's diameter, so that's like that's a respectable. That's a legit.
That's a legit size.
Yeah, that's a legit size.
I tell you, there is an even larger virus, which is 1.5 nanometers in length, or microns. 1.5 microns. You can see it in a microscope. And so...
what's it full of if it's not...
A light microscope, I mean. What's it full of?
I mean, I always think these viral genomes, does it have an enormous... I mean, yeah.
Well, some of them are large. You know, this one that I'm talking about has over a 2 million base pair double-stranded DNA genome. But there's also other space in there, and there are a lot of proteins in there, and nobody knows what they do. I mean, most of them don't look like any other protein, so we don't understand much about it.
Somebody needs to study these giant viruses, too.
Well, yes, the problem is it's hard to get funding support.
Yeah.
Right? Because it's not clear that they cause any human disease, and so there you go. That's the end of that.
That's the end of the funding stream.
But
there are some groups who do, and there are two in France that are very productive in Marseille. Interestingly, they're in the same institute, and they're deadly competitors with one another.
Oh my!
And they work on the same giant viruses. So I go to a giant virus meeting every couple of years in Germany, and they're always fighting with each other. And once I had the audacity to invite both of them onto TWiM, and a few hours before, one of them came up to me. He said, no, I can't be on the same program as that person. I cannot do it. Okay. Very good. Anyway, thank you, Petra. That was good.
Yep,
I like
thank
that.
you.
All right, now for a little virus information from Michael.
The title of today's paper is Reversible Bacteriophage Resistance by Shedding the Bacterial Cell Wall. And this paper appears in Open Biology, so it's open source. And it was authored by Angenie, Marbrook, Cruz-Roseman, Rusin, Briegel, and Clayson. And they are located at the Molecular Biotechnology Institute of Biology at Leiden University and the Center for Microbial Cell Biology, also at Leiden University, which is located in the Netherlands. So in keeping with our theme of unusual this week on TWiM, today's paper is going to describe a previously unknown but very sophisticated defense system that bacteria have employed or evolved, whatever teleology you want to use, to help them withstand an onslaught of a lytic phage attack. And recall that lytic means that the host, the bacterium, will suffer a violent and explosive death, releasing approximately 150 in the case of TWiM phages that many of us are familiar with from our work in E. coli, after about 60 minutes of incubation in the presence of phage at about 37 degrees. Now, this defense system is found in both gram-positives and gram-negative microbes, or as the authors describe them to us, the mono and diderm bacteria, to denote that they either have one unit membrane, as is the case in the gram-positives, or they have two unit membranes, as is in the case of the gram-negatives. So then why do these authors talk with us about membranes, and how are they important to our developing and understanding of this unique defensive system that protects and enables bacteria to withstand a lytic attack? Simply, the bacterium does it by shedding its shell wall. Now, recall that the bacterial cell wall serves as a molecular corset. And this example that I'm going to describe comes from Arthur Koch that I introduced you to in a few minutes ago. And he was one of the professors that taught me about cell division and peptidoglycan when I was a graduate student at IU. And what this molecular corset of peptidoglycan does is it lends form and function to the bacterial cell. And what we will learn is that in this paper, that is upon exposure to the phage, the microbe undergoes a behavioral change where they rapidly and nearly completely convert themselves from walled cells to a cell wall deficient state. Now, let's take a step back and think about this behavior for a moment. No cell wall. How could the selective pressure drive the development of what is likely a multigenic event? Now, the authors will offer that these wall-less bacteria remain completely viable insofar as they remain in an osmoprotective environment. Hence, their obsession with how many membranes the microbes have, and they are able to revert to a wall form of their former cells. Now, in their abstract, they tease us and offer that while shedding the cell will dramatically reduce the number of progeny phages produced, hence the selective pressure, it does not always preclude phage infection, as they so beautifully are going to show us when they infect Bacillus subtilis with Phe 29. So let's quickly review the process of a bacterial infection. Since this isn't TWiV, this is TWiM. So you have attachment. The virus literally has to bump in via Brownian motion because they have no motility to the bacterium. So they have to attach. So you have to have a receptor-ligand interaction. At that point in time, you have ejection of the nucleic acid from the virus into the host, at which time the viral infection enters what is referred to as the eclipse period. And it's here you have the coordinated expression of viral genes that ultimately result in the lysis of the bacterial cell. And what I found remarkable about this paper and the results that they offered was the formation of the cell wall deficient cells prevents complete eradication of the bacterial population, I should say. And what is it that Jeff Goldblum, as his character Ian Malcolm, said in that first Jurassic Park movie before the dinosaurs ate everyone? Life always finds a way, which is really saying selection operates on a population and not an individual. It's this powerful force, or the old adage from Friedrich Nietzsche, that which does not kill us makes us stronger. And I think that's what this is showing us. This paper so beautifully shows us. So here the authors frame their data elements to suggest that this remarkable behavioral adaptation, switching to a cell wall deficient phenotype, may potentially limit the efficacy of phage therapy, especially when the infection is in an osmo-privileged environment, like many of the environments that infections take place, because they're perfectly isotonic, and, the bacterium can lose its wall, as we see with the development of L-form bacteria when we put patients on buckets of beta-lactam class antibiotics. So what they did in their story in setting this up, their first experimental offerings, they show that a filamentous actinobacteria, a monoderm, or a gram-positive, and these actinobacteria, and Petra introduced us to this concept, these are streptomyces. They're remarkable for their high GC content of their genome. Also, the streptomyces have a linear rather than a circular genome, and they include many industrial significant microbes like the streptomyces and Micromonospora, where they make a diverse range of bioactive metabolites, including antibiotics, enzymes, enzyme inhibitors, signaling molecules, and even immune modulators. So these actinobacteria can switch to a cell wall deficient state after phage infection. The authors also point out, I'll toss into the show notes also a paper showing that the actinos can even do this adaptation of going cell wall-less in response to environmental stressors. So here in the first experiment, they focus on one strain of streptomyces, specifically MBT86, which was isolated from the Qinling Mountains of China. And they simply grow the microbe in an osmoprotective environment using something they term LBP medium, which contains, and I'm just going to give you the recipe because I think you'll appreciate how it's osmoprotective. Got 0.5% of glucose, not very protective, 0.5% of yeast extract that provides carbon and energy and proteins, 0.5% peptone, same thing, carbon, energy, and proteins, 20% sucrose. Now we have our osmoprotection. And then, of course, for good measure to make DNA, we have 0.1% mag sulfate to supply as a cofactor. So when the bacteria are grown in this osmoprotective LBP medium, in the presence of a Streptomyces phage, LA7, it can actually produce 10,000 plaque-forming units per milliliter in about 24 hours. 10,000 plaque-forming units. This is absolutely huge because the average T phage has a burst size of about, 150, Vincent, give or take? Mm-hmm. And the Bacillus subtilis phage that they use here, Phe29, has a burst size between 5 and 600. So this is a phage that really makes progeny. And it does it in about 24 hours. And so what they observed is that all the remaining cells exposed to the phage don't have cell walls after lysis of the dominant mycelial population. Now, since this is open source, I'm going to tell you to go to the figure and then download the PDF and then blow it up, because in panel A, you can see the mycelia. And this is the control figure. Panel A is the control. And you can see the mycelia. It looks fuzzy and out of focus when you just look at the print copy. But when you blow the sucker up, you see the mycelia. But then over on panel B, you show what the phage did. Now, you got intact membrane, you got DNA, and these suckers have no cell wall. And ask yourself what you are seeing. You are literally seeing that somehow they've turned off the ability to make peptidoglycan and confer this mycelial form, because streptomyces look like a very small version of a fungus. And it's only possible that you lose your cell wall in this osmoprotective medium. If they didn't have the osmoprotective medium, you'd get lysis, mass destruction, and everybody would be dead. And they really do this as an old-time microbiologist would do with vision. You see this beautiful layout of electron micrographs that they show you phase contrast image, and then they use Cyto9, which sees DNA, and then they give you a FM595 stain, which stains red, which shows you where the membranes are. And then they merge the files together like all good confocal microscopes do. And you can see that it's intact. The DNA is surrounded by the unit membrane, and you could potentially have a live cell. And then they do the viable count. They actually show that the cells can grow even after losing their cell wall. And so they measure it, they count the number of cells. And what they found in their quantitation experiment is the number of cell wall deficient cells after 24 hours was 34 times higher in the presence of phage, LA7, where they had about 204 plus or minus 125 cell wall defective cells, as opposed to samples without the phage, where they only had 6. So that's a 34-fold difference.
So 2 points. So 2 points. One is it's just as these are on solid medium too, not in liquid. Is that correct? Or is this liquid medium? So
well, they did some in liquid and they've done some in solid.
Okay. And then the other thing is, I think this is actually goes back to this long history, right? Of in E. coli and bacillus, there's a lot of work, some from the Arrington lab, on these L-form bacteria where you can treat them with antibiotics that do cell wall, and you select for these L forms. I think what's really cool here is, in a way, this is more natural in that these are the phage that are selecting for them. And this is obviously a totally different group of bacteria.
Yeah, the antibiotics are selecting for the L form, and the phage are selecting for this behavioral change. And selection is the king.
Yes, selection is the king. Although here it seems like there's also, because it's higher, if you're selecting, that they get a pretty high frequency of these. Do they? Yeah.
They do. And that's really the gist of the paper. I mean, they go on showing that this is a real event and that these cells can actually revert or switch back to the canonical mycelial mode of growth. So they can actually turn off the cell wall-less phenotype and go back to making mycelia. So I'm sorry to my dear colleagues and exponential phage proponents as antimicrobials. This importantly demonstrates that a considerable fraction of a population that survives a phage attack suggests that once your reticular endothelial system has cleared phage from your system, an infection has an opportunity to resume. It's sort of like that old adage, we'll be back. And
I guess so, but these are streptomyces. So are these, as far as I know, none of them are pathogens.
No. But we have this, now we go on to the next aspect of their paper. They take us to E. coli, which is a pathogen. They take us to bacillus, and they get us to jump that this is a universal behavior that seems to be conserved in the microbial world. So they did this by asking a simple question first. Is this dependent on the phage? Do the phage carry a gene product that results in the generation of a wall-less phenotype? And the simple answer is no. For when they used other lytic phages, they saw a qualitatively similar response and a marked increase in cell wall deficient cells, first in the streptomyces. And in fact, they used 3 different phage besides LA7, CE2, CE10, and LD10. And these suckers make a lot of phage. CE2 makes 30,000 PFUs per mil. CE10 makes about 2,500 PFUs per mil. And LD10 only is wimping out at 20,000 PFUs per mil. That's a lot of phage when you consider what E. coli is producing or bacillus. And so they now are asking the next question that Petra tumbled to is, what about, the medically relevance, the gram negatives or the diderms, as well as other gram positives, like the canonical gram positive microbe that everyone uses as their model system, Bacillus subtilis. So here they use 2 lytic phages, 529, again, a burst size of 570 in subtilis, and T4 for E. coli, which has a burst size about 150. And you guessed correctly. 24 hours after the addition of phage, both cultures contain cell wall deficient cells if the population was in an osmoprotective medium. And they did not appear to proliferate. And again, they do this beautifully with staining, showing us that the E. coli hasn't quite lost all of its peptidoglycan, but the Bacillus subtilis, with its only single membrane, had no peptidoglycan remaining. So the double membrane may have protected E. coli a little bit longer. And then they did a really cool experiment in their attempt to address how this cell wall deficient phenotype was occurring. Here they queried whether the formation of the cell wall deficiency was due to endolysins released by the lice bacteria. So then taking a phage-infected Bacillus subtilis culture from the osmoprotected medium, they filtered it, and only the supernatant was added to a fresh bacterial culture. So the question they're going to ask is, will this supernatant extract result in a cell wall-less behavior? And the answer is no. It did not give rise to cell wall defective cells. Now, here they're really careful, suggesting that there was either insufficient cell-free lytic agents secreted into the medium to induce this response, or it just simply didn't have it. And that's the problem with negative results. You really don't know what's actually giving you the null result. Now, in their mechanistic quests, they then asked whether artificially produced cell wall deficient cells might survive phage infection. Now, most of our introductory micro students have heard their micro professor talking about spheroplasts for gram negatives and protoplasts for gram positives, and they never thought they'd ever actually see them in a research paper. Well, here they are. And the spheroplasts and the protoplasts do not increase in number without their cell wall. And you can make these cells simply by using lysozyme or antibiotics to form, and your professor probably droned on about how creating L forms, and you wondered whether you would ever use it. But here, they're using it to try to decipher this mechanism. And what they argue here about these L forms is these results offer direct evidence that wall deficiency provides protection against lytic phages. Interesting, the phages were found near artificially produced spheroplasts of E. coli, and then they raised the question whether the phages could attach and maybe even eject their genomic material into the cell wall deficient cells. Remember, this is truly DNA ejection from the phage into the host. It's not injection, it's ejection, because it's an abiologic behavior by the phage. They then went on to another really sophisticated microscopy technique called cryo-electron tomography to address the question whether the phages could attach and maybe even eject their genomic material into wall deficient cells. And they give us some beautiful three-dimensional data of E. coli spheroplasts with phage T4, revealing a highly heterogeneous sample with an inconsistent appearance of cell wall deficient cells in outer membrane vesicles. And they give us a movie to look at. And again, this is all open source, so you can watch the movies. However, the bottom line with the E. coli, the diderm, is no phage attachment was observed for any of the cell wall deficient cells or cell fragments that they imaged. Now, in the EM data from Bacillus subtilis, Phi29 was not only able to attach to the protoplast, but could also eject its DNA into the cells, and that DNA replicated because Phi29 is a DNA phage. And the authors observed that the attached phages filled, as well as they found empty capsid heads, suggesting DNA ejection, and they also detected filaments inside some of the protoplasts that were surrounded by phages, and these filaments strongly resembled P1 proteins that are associated with P 529 DNA replication. And all of this rolls up that 529 was able to attach, eject, and even produce new phage progeny in Bacillus subtilis protoplast, as indicated by what they saw. Now, in their explanation and their discussion, they postulate that phage 29 normally binds to the glycosylated teichoic acid. And you recall from your cell wall lectures that glycosylated teichoic acid lives in peptidoglycan of gram-positives. But what phage 29 can also do is it can bind sporadically to fragments of lipoteichoic acid, which also from your cell wall lectures, is attached to the membrane. So the phage had an attachment site it could attach to. So it really drives home the fact that, life will prevail. These phage want to replicate, and in the gram positives, they can. The gram negatives are a little bit more stingy. Now, by contrast to the L forms, the cell wall deficient cells produced after a phage infection are not able to proliferate and will switch back to a wall mold of growth under favorable conditions. Bottom lining this, transiently lacking or shedding your cell wall is a common and general escape mechanism against phage attack. Now, this has created more questions than answers. Now, the exact mechanism behind this phenomena of bacterial shedding their cell wall in response to bacteriophages in an osmoprotective medium remains to be uncovered. But this is something that anyone who's really considering phage therapy really needs to think about, because many infections are found in osmoprotective environments. And so we may be lulled into a false sense of security in our labs thinking that phage therapy will work, when in reality, the bacteria and the phage have already figured this problem out and said, nope, it ain't gonna work. And so this was a really cool paper, and I thought you guys would enjoy it. It reviews cell wall biosynthesis and really drives home the fact that life will prevail. The bacteria and the viruses are in this tour de force, this constant struggle, and they have been evolving for billions of years.
So I had a couple of questions. One is, why lose your cell wall if you can still be infected?
That's only in the gram positives, yes.
So it seems like a lot if it's like...
I think it's an extreme measure.
Right. Maybe it makes you don't lyse as easily, because sometimes...
Well, you don't get as many phage. And remember, the streptomyces are principally soil organisms. And depending upon the flow of liquids, the phage may get diluted away. So you may be at that threshold where if you have a really productive lytic infection, it will infect that clot of dirt. But if you lose your cell wall, you may live long enough to survive the phage infection. Your phage get washed away by simply rain, and the phage then have to find the clot of dirt that has another streptomyces. So this behavior adaptation of losing the wall and then reverting back could be that simple of a selective pressure. You drop the phage concentration below threshold.
Or it could be because the phage lysis, I don't know how 529 works, but some of them actually, it's a cell wall. Essentially, the cell wall is damaged, and then the bacteria can't deal with trigger pressure. So they must, even with the high osmolarity, it could be that losing the cell wall is protecting them from, they can essentially serve as hosts for the phage without exploding at the end.
Yeah.
And then the other thing is, one of the things about the L forms that are selected for by using cell wall active antibiotics, like in the beta-lactams, like penicillin or in that class, the people have been wondering if they exist, and there's some evidence they might be there in some cases. Here you can see another reason L form they seem to be slightly different pathways. So that's also going to be really interesting to compare. But whether these exist all the time as sort of a bet hedging idea.
Yeah. And maybe the L form, one of the questions I had for myself is, does this validate L forms as being real? And I think that's what you were trying to ask me. Are L forms real?
Well, L forms are real, but are they physiologically relevant? Yeah.
Yes. And I think this paper effectively said they could be.
Right. I think it's adding to they could be. Right.
Yeah.
For sure.
Michael, the loss of the wall requires osmoprotective environments, and they say these may occur in nature. So where in nature would you find such an environment?
I guess any place where you have enough of these molecules like sucrose.
Mm-hmm.
You could see this osmoprotective environment associated with nectar. You could see it any place that you have these high concentrations in honey. There are a lot of environmental niches that aren't thought of all that often in terms of growing bacteria. And in fact, that's effectively the way natural products are indeed protected. They dewater themselves and increase, the osmolarity of the solution to a very high osmotic pressure.
Mm-hmm. Okay. Now, in a human being treated with phages, they say that if you happen to be giving them cell wall targeting antibiotics, it would have the same effect.
Yeah.
You don't need an osmoprotective, so that's not good.
No.
All right.
Yeah, so it's a fascinating story.
It is. And so why did it take so long to figure this out? I mean, people have been studying phages for ages, right?
I don't think anyone's ever looked.
That's it, huh? You just haven't looked.
You have to look in an osmoprotected environment, and I think that most people didn't. And the Kleisen lab they work on the streptomyces, so they were already working on the organisms themselves, and I think they've seen streptomyces L forms in other situations. So I think for them, this is sort of a natural extension.
Yeah. Okay.
But yeah, that's because of this high osmolarity. Having to add it, the physiological relevance does come up, because even in the human body, there might be parts. I mean, it's very densely packed in the intestine. It could be that E. coli in some places is somewhat protected. I don't know enough. Or maybe if you're in a biofilm and you're fixed and you're really close together with this, polysaccharides keeping everybody together, that might be enough. It's a good question.
Right. Thank you, Michael. Fascinating on both ends today. Let me read one email that we have from a colleague of Michael's. Jackie writes, I've hesitated sending this email as this is very likely very elementary, so I apologize in advance. No apologies needed for any questions. There was a recent episode of TWiM that you guys mentioned. You didn't receive many emails, so here you go. I'm a new listener and I'm loving it. Unfortunately, I do not have a formal background in micro, but I daydream of it. One day, ha, ha, ha. So far, none of my colleagues have complained about me listening to TWiM while I prepare our equipment for the day. Yay. I have an inquiry regarding ergot toxin. Random, I know. Is the toxicity and resulting clinical complications due to all ergot alkaloids or only to those species of ergot alkaloid-producing fungi that also produce sclerotia? What's different between the fungal species that result in ergotism while others do not? Side question, are sclerotia basically the fungal version of bacterial inclusion bodies, storage? I've read that this mycotoxin can cause gangrene in both humans and animals, and additionally that ergotamine is a building block of the psychoactive drug LSD. How is ergotamine different enough not to cause LSD users to develop gangrene at times, or does it? Additionally, how would a crop become initially contaminated with ergo alkaloids? If a field were to be contaminated, would the fungi now live in the soil and compromise future yields? If these mycotoxins are the same as others and are chemically stable, how would you treat an infected field? Recently, there have been numerous reports of food poisoning type illness, envy, and diarrhea, allegedly caused by Lucky Charms cereal, so far unfounded. Theoretically, could that possibly be the presenting signs, symptoms of ergotism and an ergot toxin event? Does ergotism only come from ingestion of the toxins, or can one get it from other routes? All the questions. Thank you in advance for fueling my nerd tendencies. Stay safe and keep being awesome. Okay, Michael,
All right.
Anyone.
So
I don't know.
I just put into the show notes for you to read. ASM published an article in 2018 for the lay public, From Poisoning to Pharmacy, A Tale of Two Ergots, which is a good introduction into the field, and it will address some of your questions. And I also put into the notes an article from Medical News Today that talks about ergot poisoning. And principally, these sclerota are operationally defined as the hard, dark resting body of certain fungi, consisting of a mass of hypal threads capable of remaining dormant for long periods of time. And typically what happens when ergots begin to grow, the fungi begins to grow on the grains and actually produces the ergot toxin, it's simply because the grain has been improperly stored or has gotten wet and the fungi can begin to grow. I wish Elio was here and he would have much more eloquent ways of describing it. But this is a well-known phenomenon that this hallucinogenic toxins from the fungi could actually be imparted to grains, and they would actually then go into some of the foods that we would eat. And St. Anthony's fire was a hallucinogen that was associated with rye fungi or the contamination of rye grain with the fungus. And then they were also the Salem witch trials were blamed on improperly stored grain and this fungus, resulting in all of these bad things. As to whether or not these mycotoxins can cause gangrene, I just don't know enough about mycotoxins, and I'm hoping someone else in our group knows a little bit more. And I just apologize I didn't have time to go after it. In terms of the LSD, when I took organic chemistry in the dark ages, we actually had to make LSD. And so ergotamine is a precursor, and LSD is, of course, a synthetic derivative, and it was all part of our organic chemistry wonder. And of course, they immediately confiscated the product because we weren't too far away from the 60s to understand all of the wonders that LSD did.
We just had to make fake strawberry flavor.
You just had to make fake strawberry? Yes.
I guess they didn't trust us even with the precursor. So.
Yes. Yeah, we made two things. We made LSD and cocaine.
Wow.
The trick was not to make strychnine, and because if you screw up the synthesis, you make strychnine.
That's bad.
Yeah.
Where did you do this, Michael?
When I was an undergraduate at University of Illinois.
Illinois.
Yeah,
we were just trusted with fake fruit.
Yeah, fake fruit. Well, those are all very important. Imitation flavors are a huge business.
Yeah.
As to Lucky Charms, I would just, it's all about how good the grains are and whether or not they're contaminated. And some of these toxins are heat stable, and Elio would know better than me which ones are more heat stable than the others. But there's nothing wrong with your nerd tendencies. All of this stuff makes sense, especially when you live in a wet climate like Charleston.
Thank you, Michael. Appreciate that. All right, that's it for TWiM 269. Show notes at microbe.tv/TWiM. If you like, if you'd like to send us a question or a comment, you could send them to twim@microbe.tv. And if you like our work, consider supporting us. Go to microbe.tv/contribute. Earlier this week, I got an email from a listener who said, "Hey, there hasn't been a TWiM since June 25th. Is TWiM dead?"
Oh,
I'm like, no, why would you think it's dead? Anyway, I said one is dropping this week. We just decided to delay it because the one from ASM only dropped last week, I think.
Right.
So, but we're still here. We're not dead, but we're glad you're interested and keep listening. Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And Petra Levins at Washington University in St. Louis. Thank you, Petra.
Thanks, everybody.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.